Hello everyone, I am here to explain molecular biology based on the textbook Molecular Biology of the Gene by James D. Watson, Tanya A. Baker, Stephen S. Bell, Alexander Gann, Michael Levin, and Richard Lucifer. I felt that in many competitive exams like CSIR, there will be questions based on this textbook. First of all, I shall explain the contents of this book. This book contains 5 parts. Part 1 Chemistry and Genetics, with chapters like the Mendelian view of the world, nucleic acid convey genetic information, importance of weak chemical interaction, importance of high energy bonds, weak and strong bonds determine macromolecular structure, and the next part is maintenance of the genome, with chapters like the structure of DNA and RNA, genome structure, chromatin and nucleosome, replication of DNA, the mutability and repair of DNA, and homologous recombination at the molecular level, site-specific recombination and transposition of DNA. And the next part is expression of the genome. Chapters like mechanism of transcription, RNA splicing, translation, the genetic code. Next, part 4 is regulation, that include chapters like transcriptional regulation in prokaryotes, transcriptional regulation in eukaryotes, regulatory RNA, gene regulation in development and evolution, genome analysis, and system biology. And the last part is method, with 2 chapters, the technique of molecular biology, model organism. In chapter 1 of part 1, they have explained important theories and findings that help the scientific community to find explanations to many unanswered questions. Part 1, that is chemistry and genetics. The first chapter is the Mendelian view of the world. Human beings are unique among living organisms. We have developed complex languages that allow meaningful and complex interplay of ideas and emotions. Great civilizations have developed and changed our world's environment in ways inconceivable for any other forms of life. So, something special differentiates human from every other species. Then, we seek the origin of and explore the reason for our existence. Later, it seemed natural to think that human species and all other forms of life must have created at a fixed moment. This belief was questioned when Charles Darwin and Alfred R. Wallace proposed their theories of evolution. According to this, According to this theory, they stated that the various forms of life are not constant, but continually give rise to slightly different organisms, some of which adapt to survive and multiply more efficiently or effectively. At that time, they did not know the origin of continuous variation. So, the immediate consequences of Darwin's theories was that first, it helped in the realization that life first existed on Earth more than 4 billion years ago in simple forms like bacteria. So, the essence of the living state is found in every small organism. Second was the basic principle of life applied to all living forms. Then coming to Mendel's discoveries. First is the principle of independent segregation. This can be considered as Mendel's first law. According to this law, the dominant gene, that is, the trait appeared in F1 progeny, and the recessive gene, that is, the trait does not appear in the F1 progeny, will be transmitted independently without modification or mixing, and so are able to segregate independently during sex cell formation. In the picture, we can see that there are two parental generations, one with round seed and the other with wrinkled seed. The genotype of round-seeded plant is Capitula capitula, and the genotype of the plant which produces wrinkled seed is smaller. On gamete formation, each of these parents are able to produce only a single type of gamete that bear a single type of allele. That is, the gamete bearing capitula in the case of plant which is able to produce round seeds, and smaller gametes with allele, smaller in the case of plants which are able to produce seeds with wrinkled seeds. Okay. Upon fusion of these two gametes, definitely there will develop a hybrid. Okay, so in the F1 generation, they obtained a hybrid with a capitula allele and a smaller allele. Upon selfing of this hybrids of F1 generation, here we will get two types of gametes from each parent. That is, one gamete bear a capitula and the other gamete bear a smaller. Similarly, the other parent also produce two types of gametes with one gamete with capitula and the other gamete with smaller. And upon fusion, each and every gamete which bear a single copy of this allele have the potentiality to fuse with any of these gametes. So altogether, there are obtained three types of genotypes and two types of phenotypes. The plant which produces a round-seeded variety or round seeds are able to produce, or they are having two types of genotypes. That is, some of them bear capitula and the others bear capitula smaller. And the plant which are able to produce wrinkled seeds having the genotype smaller. So here you can see that the gametes did not get modified or did not change at all. So during gamete formation, each and every individual alleles have their capacity to move into a gamete, and they can freely fuse with the other gamete without any modification or changes. Okay. But some alleles are neither dominant nor recessive. Example is pure breeding red snapdragon, that is Andirrhinum. Andirrhinum is the scientific name of snapdragon. And pure breeding white snapdragon gives F1 progeny of an intermediate pink. In this picture, they have shown the inheritance pattern of snapdragon, that is Andirrhinum. Here, the parental populations are having one with red flower and the other with white flower. The genotype of the plant with red flower is capital A, and the genotype of this white flower bearing plant is small a. And similarly, just like that of the first case, here also they were able to produce a single type of gametes because they are pure breeders. Single type of gametes during gamete formation, and when these gametes get fused, they develop the F1 generation. Definitely, we know that F1 will be a hybrid. So, in this hybrid condition, that is, combination of capital A and small a will give intermediate pink color. Okay, so when this F1 progeny gets set, each of these parents are able to produce two types of gamete, capital A and small. Gamete with capital A and gamete with small. So upon fusion of the gametes, they are obtained three types of phenotypes and three types of genotypes. Here, the phenotypes are red with genotype capital A. Second phenotype intermediate pink color with genotype capital small, and the white flower bearing plants with genotype small. Here also there is no mixing at all, but sometimes the alleles do not show the dominant recessive pattern. So with this example, they have explained that concept. Then coming to the second principle, that is principle of independent assortment by Mendel. And it is considered as Mendel's second law. According to this law, each gene pair independently transmit to gamete during sexual formation. Any one gamete contains only one type of allele from each gene pair. There is no tendency of genes arising from one parent to stay together. So in this picture, they have tried to explain the second concept, that is the principle of independent assortment. Here, a plant with round yellow seeds and a plant with wrinkled green seeds were considered as the parent generation. Here there are two genes, here also two genes are there. And the genotype of plant with round yellow seeds was capital R, and capital Y, capital Y. At the same time, the genotype of the plant which bear wrinkled green seeds produce are having the genotype small y, small. Upon gamete formation, each of these genes, here there are two gene pairs. Here also two gene pairs are there. From each gene pair, one allele will move into the gametes. Here also, one allele from each gene pair moves to gamete. Upon fusion, there arise F1 generation, that is definitely a hybrid with the genotype capital R, capital Y, small y. And upon selfing of this F1 generation, there, there develop four types of gametes from each parent. Capital R, capital Y bearing gametes, capital R, small y bearing gametes, small r, capital Y bearing gametes, small r, small y bearing gametes. Okay. So from this picture or from this description, they try to explain that each allele of each gene pair show a tendency to move towards the gametes. One allele of each gene pair move towards the gamete without any interference, and they do not show tendency to stay together during gamete formation. Okay, that means this capital R, small r, capital Y, small y. Here, this capital R have the potentiality, or they did not show any tendency to stay with capital Y or small y. So the gametes along with the gametes which pair single copy of each allele, each gene pair. Okay. Next comes the chromosome theory of heredity. The principal reason for the original failure to appreciate Mendel's discovery was the absence of firm facts about the behavior of chromosomes during meiosis and mitosis. So this was the major reason behind the original failure to appreciate Mendel's discovery, because at that time there was no idea regarding the behavior of chromosome during meiosis and mitosis. So Mendel's laws was confirmed by the classic paper, The Chromosome Theory in Heredity, by Walter S. Sutton in 1903. According to Sutton, the diploid chromosome group consists of two morphologically similar sets, and that during meiosis, every gamete receives only one chromosome of each homologous pair. He then used this fact to explain Mendel's result by assuming that genes are parts of the chromosome. Then coming to the next topic, gene linkage and crossing over. Mendel's principle of independent assortment is based on the fact that genes located on different chromosomes behave independently during meiosis. So, the Mendel's principle of independent assortment is based on the fact that genes located on different chromosomes, that is why they behave independently during meiosis. Often, however, two genes does not assort independently because they are located on the same chromosome. That is, the genes which are located on the same chromosome are linked. Okay, so the linked genes did not show any tendency to get separate. The number of linked groups was identical to the haploid chromosome number. That is, four groups of linked genes in Drosophila and four morphologically distinct chromosomes in haploid. So we can say that the number of linked genes or linkage groups in a particular organism will be based on the number of number of haploid chromosomes or number of haploid chromosomes. Okay. For example, four groups of linked genes can be seen in Drosophila because they are having four morphologically distinct chromosomes in a haploid cell. Linkage, however, in effect, never complete. The probability that two genes on the same chromosome will remain together during meiosis ranges from less than 100% to nearly 50%. So, the probability of these two genes in the same chromosome to stay together during meiosis that ranges from 100% to nearby 50%. This variation in linkage suggests that there must be some mechanism for exchanging genes on the homologous chromosome called crossing over. So, if there was no crossing over, we can say that the probability of two genes of the same chromosome to stay together on gamete formation is 100%. But in practice, we can see that the probability of two genes to stay in the same chromosome is only 100% to, or it ranges from less than 100% to 50% because of the phenomenon of crossing over. Crossing over is nothing but the exchange of parts of chromosomes between homologous chromosomes during meiosis. At the start of meiosis, through the process of synapse, the homologous chromosome forms pairs with their long axis parallel. At this stage, each chromosome duplicated to form four chromatids. Sorry, two chromatids. So altogether four chromatids are there. That is synapse pairing together four chromatids. That stage is called as a tetrad, which coil about one another. In this figure itself, they have shown the Janssen's hypothesis of crossing over. This picture shows the Janssen's hypothesis of crossing over. Here, first of all, synapses of duplicated chromosome form tetrads. These are two homologous chromosomes with two chromatids, and each of them forms or they will align parallelly along their long axis, okay, and form a tetrad. And this tetrad, two chromatids bend across each other. In this tetrad, two or more chromatids bend, bend across each other, and later on, a part of this chromosome will be exchanged. So each chromatid breaks at a point of contact and fuses with the portion of the other. So each chromosome gets separated with a part of the other counterpart. Okay. So this is the explanation of the Janssen's hypothesis of crossing. Then Thomas Hunt Morgan and his students proposed that genes are located close together on the chromosome would assort with one another much more regularly, that is, close linkage, than genes located far apart from the chromosome. That means the genes which are located nearby in a chromosome show more tendency to move towards the gametes They show more tendency to stay together during their passage into the next generation. But the genes which are located far apart on the same chromosome will show less tendency to move together into the next generation or next gamete. So they use this way to locate or map the relative position of genes on the chromosome and produced a genetic map. So, as they said, as T.H. Morgan and their students proposed that the genes which are close together in a chromosome show a tendency to move together into the gametes during meiosis. But the genes which are located far apart on the same chromosome show less tendency to move together into the gametes. So they use this concept to locate or position or map the genes on the chromosome. They used the frequencies of various recombination classes or recombinant classes. Okay. They used the frequencies of various recombinant classes to locate or map the genes on the chromosome. Consider the segregation of three genes located on the same chromosome. Suppose there are three genes which are located on the same chromosome. The arrangement of genes can be determined by means of three crosses, in each of which two genes are followed. So, two factor crosses will be made. Suppose a cross between capital A, capital B and small b yield four progeny types. That is, two parental genotypes, that is capital A, capital B and small b. And two recombinant types, that is capital A, small b and small a, capital B, that is recombinant type. Okay, so at a time they will consider only two genes at a time. So that is called as two factor cross. In order to find out the location of or the position of three genes on a chromosome, they carried out 2-point cross or two factor cross. Okay. So that in the next cross they made they cross between capital A, capital C and small a, small c. So the second time they consider two genes at a time, that is capital A, capital C and small a, small c. Okay. So similarly, this cross also gives two parental combinations as well as the recombinants, that is two parental combinations are capital A, capital C and small a, small c. But the recombinants was capital A, small c and small a, capital C as recombinant. So a cross between third they made cross between or considering capital B, capital C and small b, small c. At that time they obtained parental types and recombinants. The parental types were capital B, capital C and small b, small c. But the recombinants were capital B, small c and small b, capital C. Each cross will produce a specific ratio of parent to recombinant protein. Okay, so there was during each cross they found there was a specific ratio of parent to recombinant protein. Okay, for the first at the time of first cross, that is the cross between cross by considering two genes, that is capital A, capital B and small b, they got 30% recombinant and 70% parental type. The second cross by considering capital A, capital C and small c, they got 10% recombinant and 90% parental type. On the third cross, that is the cross between capital B, capital C and small b, small c, they got 25% recombinant and 74% parent. And from this result, from this specific ratio they obtained, specific ratio of recombinant and parent that they obtained during their experiments, tells that genes A and C are close together. Genes A and C are close together than A and B and B and C. Because at the second cross, that means the cross between capital A, capital C and small c, they obtain only 10% recombinance. The remaining 90% was parental type. As I said earlier, the genes which are closely located show less tendency to get separated during gamete formation, or they show more tendency to stay together during the gamete formation. So here in this picture you can see that uh, here they have shown it in the picture. Here the A and C, the recombinance of A and C obtained only by 10%, and the recombinance of B and C 24%. Okay, so from this picture, from this description, we can say that the distance between A and C are less, that is 10%. At the same time, the A and B and C are, the distance between A and B and C are somewhat similar. So the gene order will be A, C, B. A, C, B will be the gene order. That means A and C are more closely together because of the decreased rate of recombinance, or the decreased ratio of recombinance. And the B and C are 25% because the rate B and C are 25%. Distance between B and C are 25%. Okay. So the, the accurate mapping is often disturbed by the phenomenon called as interference. So there is a phenomenon called as interference that will inhibit the crossing over to happen to nearby locations. Okay, so one crossing over will inhibit the occurrence of another crossing over on its vicinity. That is called as interference. So the accurate mapping is often disturbed by the phenomenon called as interference. And using such reasoning, Morgan and team, that is D.H. Morgan and his team, could assign the location of more than 85 mutant genes in Drosophila. The distance between genes on such a map are measured in map units. So the distance in the genes, or the distance between the genes on the such type of maps are measured in map units, which are related to the frequency of recombination between the that is, if the frequency of recombination between two genes is found to be 5%, the genes are said to be separated by 5 map units. So based on the frequency of recombination, we can say the distance between two genes. But there is high probability of double crossover between widely spaced genes. But there is another problem. There is a high probability of double crossover. Two crossover will happen, okay, between widely spaced genes. So such assignment of map units will be accurate only if recombination between closely spaced genes is followed. Such type of assignment of map units will be accurate only if recombination between closely spaced genes are followed. The two genes will be separated if an odd number of chromosomes occur between them. That means if there happens odd number of crossing overs, then we can expect non-parental combinations. But they will end up together if an even number occur between them. That means if there is an even number of crossing over, we cannot expect, or the, the parental type of progeny will be more enough. Okay, that is what I wanted to tell you. Then, okay, so the two genes will be separated if odd number of crossing over occur between them. But they will end up with or they will end up together if an even number occur between them. Here, this picture, they have shown that there are three genes, capital A, capital C, capital B, small a, small b, small a, small c, small d, the pattern generation. Okay. And on gamete formation, capital A, capital C, capital B bearing chromosome will go to a one gamete, and the small a, small c, small b bearing bearing gametes will there will be a type of gamete which pair small c, small a, and small b combination. Okay, if crossing over happen between capital A and capital C, we will get the gametes like capital A, small c, and small b, because the recombination happened between capital A and capital C, or in between this A and C. So the gametes, the recombinant gametes like capital A, small c, small b, and small a, capital C, capital A will be produced. Here, if the crossing over happen between capital C and capital B, then the result will be like this: capital A, capital C, small b, small a, small c, capital A. Okay. So, okay. Then coming to the double crossover. Here, even number. These are examples for the odd number of chromosomes. Here you can see double crossover happens. That is even number of chromosomes. Here the crossing over happen between A and C and C and B. So, so what will be the result? Capital A, small c, capital B. The result, and small a, capital C, and small. So at the end, here we are considering capital A and capital B. By this double crossover, they remain together because there occurred even number of crossing over. But here during odd number of crossing over, there happened the genes get separated. But here, if you are conducting a cross by considering two genes at a time, capital A and capital B, we could not say there happened crossing over because they remain together. They remain together during the gamete formation because of the double cross. So in 1950, more than with his students published their books, The Mechanism of Mendelian Health. So, coming to the next part, the origin of genetic variability through mutation. Now we know that the hereditary variation forms the basis of the theory of evolution. So, we know that the hereditary variation forms the basis of theory of evolution. Relatively low mutation rate together with only mild selective advantage would be sufficient to allow the gradual accumulation of new favorable attributes. So, the theory has to be modified in in this way that relatively low mutation rate together with only mild selective advantage. Low mutation rate together with only mild selective advantages would be sufficient to allow the gradual accumulation of favorable attributes and thereby evolution occurs. So then there arises various speculations about uh, what genes are and how they act, etc. But the preliminary attempt to uh, find a gene protein relationship was done by that was put forward by a gene enzyme relationship suggested by Archibald E. Garrett based on the studies of inborn errors in metabolism. So at that time there are different speculations arised. What are these genes? How they act? Okay, and the preliminary attempt to find an answer to this question was a gene enzyme hypothesis or a gene enzyme relationship suggested by Archibald E. Garrett based on studies of inborn errors in metabolism. That was extended by the studies of many scientists. However, the lack of fundamental knowledge about the structure of relevant enzymes ruled out the deeper examination of the gene-enzyme relationship. At that time, the detailed structure of enzyme was not available. So that was the reason. Because of this lack of knowledge regarding the relevant structure of relevant enzymes, ruled out the deeper examination of gene-enzyme. By that, the chapter 1 of part 1 is finished, and we will meet again with the second chapter of part 1, that is nucleic acid convey genetic information. Thank you.